Oh, ooh, okay. Hopefully our tech issues are at an end. So we're gonna finish up talking about our regulation of mean arterial pressure. Okay. So last time when we were talking, drawing stuff in pencil, the main thing we were going over was talking about how our nervous system regulates mean arterial pressure. So we were talking about effects of the parasympathetic nervous system having a mild effect because of their effect on the heart rate, but largely seeing effects of the sympathetic nervous system acting on our many factors that influence mean arterial pressure. So we were walking through this negative feedback loop called the baroreceptor reflex, which is a reaction to a sudden drop in blood pressure, where we have sympathetic activity increasing at the same time as parasympathetic activity is falling. And overall, we have the effect of our autonomic nervous system, increasing heart rate, stroke volume, and TPR are three factors that make up mean arterial pressure, raising that mean arterial pressure back up and resetting us to our set point. And that was the big picture of what we were doing last time. So that's for the short-term regulation of blood pressure. For long-term regulation, we're thinking about minutes to days. This isn't going to act really through the nervous system so much as it is going to act through our kidneys. Does someone call on do not disturb? Okay. Okay. Anyway, so long-term regulation is going to be your kidneys, and we're going to need to use hormones to do that, and we're going to need to think about blood volume to do that. So we'll talk a bit about blood volume in the kidneys when we get to our renal unit. So we have a couple of additional factors that we are going to mention, though, that relate to our mean arterial pressure so that we have a full picture here. Okay. So the hormones that we want to be aware right now that have an effect on our mean arterial pressure are epinephrine, vasopressin, and angiotensin 2. So epinephrine we've kind of talked about before, because we were talking about how it's related to the sympathetic nervous system, right? Because we have the sympathetic nervous system going to the adrenal medulla and releasing epinephrine as a result, okay? And we talked about how epinephrine binds to beta receptors in the heart and skeletal muscle when it's at low levels, rerouting more blood to those organ systems. At the same time, as it gets up to higher levels, it binds to the more common alpha receptors throughout the body, keeping our total peripheral resistance up. So that's our first bullet point here, our total effect of epinephrine. There are more alpha receptors in your body than there are these beta receptors. We're thinking more organ systems. So our total effect of all that as we are altering resistance of arterioles is to increase total peripheral resistance. So we increase total peripheral resistance, we increase mean arterial pressure. Smooth muscle effects are similar. So we have alpha receptors in smooth muscle. Those ones cause a special form of slight constriction. So we're increasing venomotor tone, which is decreasing our compliance. Overall, this has the effect of returning more blood to the heart and increasing stroke volume, increasing our mean arterial pressure. Finally, we have effects of epinephrine on the heart itself, separate from those arterioles and the veins. So we have connections directly to the SA node and the AV node, as well as to the myocardium itself. So on the heart end, when we have epinephrine binding to those receptors, we increase heart rate, increasing mean arterial pressure. We increase stroke volume, increasing mean arterial pressure. We're really focused in this unit on increasing mean arterial pressure. Not going to say that we never lower it, but most of these systems that we're focusing on, their effect is to increase mean arterial pressure rather than decrease, because that drop in blood pressure is a more kind of immediate problem for your body. So vasopressin and angiotensin 2 also increase mean arterial pressure. So these two hormones act as vasoconstrictors throughout your body. So they're causing blood vessels to constrict. We know that when we decrease a radius, we're increasing resistance. So we are increasing our total peripheral resistance, because that's just adding up all the resistance in your blood vessels. We increase total peripheral resistance, we increase mean arterial pressure. So these hormones, we'll come back a little bit also when we talk about the renal system. Okay. So when we were thinking about mean arterial pressure and the baroreceptor reflex and all that stuff, we were mainly thinking about our sensors in our arteries, so in the carotid sinus, which is up in your neck, where your internal and external carotid split from each other. You'll see it in lab eventually. And in the arch of the aorta itself. So those were in the arteries, especially in the aorta is what we're really trying to figure out pressure when we're thinking about mean arterial pressure. So those were the ones that were sensing these drops in pressure triggering the baroreceptor reflex. But they're not the only place that we have receptors that sense types of pressure, okay? So we also do have cardiac baroreceptors and venous baroreceptors, which we can kind of lump together. So both of these are low pressure baroreceptors, by which we really mean is their job is to sense volume, really, more than it is to sense pressure. Pressure is kind of an indirect measure of volume. If you're looking in, say, a vein or one of the atria, because of that relationship between volume and pressure that we saw on that compliance graph, right? There's a relationship between volume and pressure. Okay, so these receptors in the heart and in the veins, they're trying to sense volume because blood volume does overall matter to your system. Okay, so we're going to find them in the walls of big veins, so large systemic veins, and in the walls of the atria, so right atrium collecting blood returning to the heart, left atrium collecting blood returning to the heart, but from the lungs specifically. So when we see a decrease in blood volume, right, so we see less blood overall going through your system, less blood coming into the atria, less blood running through those large veins as they run into the heart. This indicates we have a problem that is related to blood pressure as well, because it's the volume of blood pushing on the walls of the arteries that creates that mean arterial pressure. So we have responses that get activated here that aren't explicitly part of the baroreceptor reflex. So that was part of the nervous system, autonomic nervous system, controlling blood pressure in response to the arterial baroreceptors. But this is the way we could get all those hormones that affect mean arterial pressure, because they affect blood volume, kind of working in conjunction here. Because we do eventually want long-term regulation to come into play if this is a blood volume problem. Okay. We got some grab bag cardiovascular regulatory processes here that also have some effects on our mean arterial pressure. So we're going to talk first about our respiratory sinus arrhythmia. Okay. So this doesn't happen to everyone, and I think it's more common in children, but in some people, we see changes to heart rate depending on your breathing, like depending on where you are in your breath cycle. Don't really know why, what the purpose of this might be, but you can try and feel it in yourself, see if it happens to you. So what happens in this respiratory sinus arrhythmia? So arrhythmia is anytime we have like a variation in the rhythm. When you breathe in, this, for some reason, is activating your sympathetic nervous system, which means that we start to have an increase in heart rate when you breathe in. But when you breathe out, that upregulates our parasympathetic branch of the nervous system. So heart rate dips. So sometimes people can use this to like steady their hands, because you're actually slowing your pulse, and so slowing some kind of movement through your limbs if you're trying to aim really well or something, if you're an archer if you're out hunting or something. You'd want to be expiring if you're trying to keep your hands steady. So because they change heart rate, this could also, in the very short term, affect our mean arterial pressure, although in the long term, this should average out. Another thing that's going to have an influence on your mean arterial pressure is something called the chemoreceptor reflex. So we'll think more about chemoreceptors when we think about our respiratory system. But one of the things that you're trying to regulate with your breathing is your carbon dioxide levels in the blood, specifically, especially. We usually focus on oxygen, thinking about how tissues need oxygen, but actually removing this waste product is really particularly critical to your body because carbon dioxide has an effect on blood pH. And if you mess up your blood pH, that's a real critical problem. So we have these chemoreceptors in the brain. They sense a number of things, but particularly they sense CO2 concentration. So if you've got too much CO2 in your blood, your body is going to change some stuff up to try and limit that CO2 buildup and breathe it out if you can. So we're going to change some of our ventilation rates. We'll talk about that when we talk about the respiratory system in response to this increase in carbon dioxide. You're going to try to fix that. But another thing that we're going to do is kind of counterintuitive. Your body's actually going to slow down your heart when we see this CO2 build up. Okay. So from one perspective, if we were focusing on oxygen, that might not make sense. You still want oxygen to tissue, you still want blood flow. But the reason we think this is the case is that your heart itself actually does a lot of metabolism. It needs a lot of ATP to contract. It's doing it all the time. So by slowing down your heart rate, one of the things you're doing is kind of slowing down the production of CO2. Okay. So we see a decrease in heart rate when we have high levels of CO2 here, partially to stop that CO2 from building up in the blood. Now, a decrease in heart rate on its own would mean that we would have a decrease in mean arterial pressure. That wouldn't necessarily be great in this situation, right? So we also have effects responding to those chemoreceptors in the brain that increase your total peripheral resistance. So this is the one time that I can think of, and one exception to what I told you, that whenever we have any one of our factors for mean arterial pressure increasing, they tend to all increase if there's an effect. Right, so this is the one kind of nitty-gritty example that we're thinking about where we actually have two opposite effects going on at once. So here we have a decreasing heart rate, but we have a larger increase in total peripheral resistance, keeping your blood pressure up in this situation, even with that slower heart rate. Okay. Another fun fact. When we think about homeostasis, keeping your body the right temperature is actually kind of more important to your survival, or at least from your body's perspective, than protecting the appropriate blood pressure. So when we have our various mechanisms for maintaining core body temperature in play, when we're trying to make sure that we're not getting too hot specifically, the thermoregulatory responses to increasing body temperature can kind of take over and drop down your mean arterial pressure, potentially to the point that you get like heat stroke, feel lightheaded, feel kind of sick. Okay. So thermoregulation is controlled by the hypothalamus for the most part. And what's happening when you're doing this thermoregulation? Say you're out in the hot, hot sun, it's summer, it's not today. Your body temperature is increasing. Okay. Our typical response to this, right, is to start sweating. So what is happening when you sweat right? You're trying to get evaporative cooling. So the purpose of sweat, of liquid on your skin, is to have that evaporate and pull some heat away from your body. In order to do that, we need to actually dilate blood vessels to the skin. So in order to get enough blood to the skin so that that blood is near the skin getting cooled off as your sweat evaporates, we see a decrease in sympathetic activity to those blood vessels that go to the skin specifically. Sympathetics constrict. So when we decrease the sympathetics, we see some vasodilation. So we're going to increase our heat loss because that hot blood is now near your skin and evaporation is happening on the skin. Okay. But if we vasodilated, right, vasodilation, we made our radius larger, right? We have just decreased our resistance, right? So we've just decreased our total number we're going to get to if we add up all the resistance in the body, because now all these blood vessels near the skin, they're extra wide. So they got lower resistance. We don't have as much kind of affecting our pressure, affecting cardiac output, things like that. Okay. So in this case, thermoregulation is happening. It's working well. The baroreceptor reflex may come into play and try and keep things steady. But if we're in a situation where we really, really have to have a lot of heat loss, we're really hot, we're really overheating, the thermoregulation is more important. So the baroreceptor reflex is not going to be able to compensate for this. This is a really strong response. So a possible consequence in this situation is that due to that decrease in total peripheral resistance, you have decrease in mean arterial pressure. You may start to feel lightheaded. You may start to feel some of those effects of that dropping blood pressure. So finally, we have a chart here of some changes during exercise. I don't really have particular comments about this. Different types of exercise have different changes here. And we can see overall how we're kind of affecting different factors as we increase exercise. So we can see that when you're exercising, we're increasing blood flow to our muscles. We've talked about how that works with our beta receptors. We are increasing our cardiac output. We know that we are increasing heart rate, stroke volume. That's why we have cardiac output increasing. We see slight increases in mean arterial pressure during this time, as well as our end diastolic volume. We know that's partially from the venous return from that skeletal muscle pump working to return more blood to the heart, stretching the heart a little, helping us increase that stroke volume and cardiac output. We do sometimes see some drop in total peripheral resistance here, but it's made up for by some of these other factors. Okay, so we are going to move on to talking about the blood that's flowing through the vasculature next. So this will probably spill over into our next unit. I expect that we'll begin the next unit with the clotting section of this packet, but we are going to talk a little bit about some of our blood cells right now. Okay. So we want to understand what is actually flowing through these blood vessels. So we're going to overview what makes up your blood. We're going to talk a little bit about the plasma, and we're going to talk about our different types of blood cells. Our erythrocytes are our red blood cells. Leukocytes are our white blood cells. Then our next topic will be talking about primarily the different steps of forming blood clots and kind of healing after injuries, and how we maintain blood volume even when we get a cut. So, really. So to start us off, as we think about what's in the blood, I want you to predict, remember from your reading, logic out, take a guess at what most of your blood is. If we do a blood draw and just leave that whole blood sitting in a vial, what's most of it going to be? Okay, let's take a look. Okay, cool. Yes, so half of you predicted correctly. So the largest portion of your blood by percentage is plasma. So our special type of extracellular fluid that we talked about way at the beginning of A&P 1 as we were talking about the different body compartments, different types of fluids that we have in the body. So I'm going to ask you a question about that plasma that is making up most of our blood. I'm going to ask you, which of these things do you think is making up most of your plasma? Yeah, let's take a look. Okay. Yeah, we talked about it when we talked about body compartments and total body water, because it's mostly water. It's extracellular fluid. It's mostly water. When we were talking about net filtration pressure, we mentioned that it does have other things dissolved in it. Yep. So doesn't have more of those other things than it has water in it. But here we have two proteins that happen to be in the plasma. Albumin is actually the more common protein that we have in plasma. It does things like carry medications and stuff around in your blood. Those proteins are important because we're remembering with the net filtration pressure that those proteins in the plasma, part of our forces for absorption, right? They pull fluid towards themselves, but they're not most of our plasma. Okay, cool. So now we're going to look at an actual blood vial and go over this in words. All right. So on average, we're thinking about how much blood you have in your body. Most women on average have about 5 liters. Men tend to have a little more blood, 5.5 liters. Right? And if we were to take a blood draw, end up with a vial of blood, we might end up with something like this as we let it settle and separate out the components. Okay. So the majority, by a little bit, of this blood is going to be made up of plasma. So that's the yellow part over here. Okay. So about 3 liters of your blood is plasma. Right. The other big component of the blood are the red parts here. So our red blood cells, our erythrocytes, make up about 45% of our whole blood volume. So not quite half. We do sometimes measure the amount of red blood cells we have in the blood. That can be important for a variety of things because we're going to see our red blood cells are carrying around gases in our blood. They're pretty important from a functional perspective. So if we measure that red blood cell content, we're measuring something called the hematocrit. And a typical level is going to vary a little bit. So they both center around 45-ish%, but you can see that there's a slightly higher range for men that's typical, slightly lower range for women that's typical. Okay. A tiny percentage of your blood would be found in this little region that separates out separately, called the buffy coat. So you can see here that if you let blood sit in a vial, spun it around to separate out the components, this buffy coat would lie between the plasma and the erythrocytes, those red blood cells. So it's about 1% of your whole blood volume. And that's where we'll find our leukocytes, our white blood cells, so immune function, and platelets, which are going to be important for coagulation, clotting, for actually healing cuts, things like that. That's most of what's in the blood. We also have this chart here of different parts, different types of blood cells and how we get to them. Okay. I'm not going to expect you to memorize this, but I do want to point out that all of our blood cells are originally starting out from these stem cells that are ready to make all our many types of blood cells, plus our platelets, because we're going to group those together here. So our hematopoietic stem cells could become any type of blood cell, but as they start to divide and start to differentiate, we're going to go down different pathways, right? So we are going to see that we have one big pathway over here, right, that leads us to a variety of white blood cells, okay, as well as our platelets and our red blood cells, our erythrocytes, and a special kind of flow that gets us to our final type of white blood cell over here. I'm going to point out to you, we are mostly going to start out by talking about red blood cells. So I'm going to point out to you how different some of these blood cells look, right? So I want you to notice that our little mini red blood cell over here, our little erythrocyte, just this tiny little disc, doesn't have all these parts inside like our white blood cells do. But we can see that as we were producing it, once upon a time it did, right? Once upon a time it had like a nucleus, other stuff going on in the cell. But by the time we get to a mature red blood cell, we've lost a bunch of components of a typical cell here. And that's going to be relevant to how and why we cycle red blood cells through our body. Similar thing is happening over here on the platelet end, but we'll talk about those later on. Similar but different. Okay. So our plasma, 90% water, mostly water, 6 to 8% proteins, giving us that capillary osmotic oncotic pressure when we're thinking about net filtration pressure. It's got big concentrations of some ions like sodium, also chloride ions. We think a lot more about sodium when we think about how our different organ systems function as we're stepping through this unit. Happens to be the case that chloride ions are also high. Kind of makes sense because sodium chloride is like table salt, right? So you would eat them together. We have lower concentrations of some other electrolytes, some other ions, but they are still important. So we have hydrogen ions or protons. We have bicarbonate ions. We'll be thinking about those when we think about blood pH in particular. We've talked about why potassium is important, why calcium is important. We know these play a role in the function of the nervous system, as well as in the function of muscles and the heart, all sorts of stuff. Okay. So we're going to move on from the plasma to those erythrocytes that I was pointing out. So you have about 5 billion of them per milliliter of blood. Got lots and lots of red blood cells. They're pretty small. They have to get all the way into all your capillaries and stuff. And they have a specific shape to them, right? So they look kind of like a donut if you're looking from the top, but that donut is filled with a little flat part in the middle. So we described this shape as a biconcave disc. So it's a disc, it's a little frisbee, but it's got 2 chunks kind of bit out of it. That's the concave part, bi because it's both on the top and the bottom. Okay. The effect of this is that we're maximizing our surface area, or at least we're creating a really large surface area across the surface of our red blood cell, minimizing kind of how much stuff is inside, right? So if we had a typical cell, right, kind of spherical, our generic cell that we draw, you'd have a lot more kind of space inside and slightly less surface area here. Okay. This is going to be really helpful for us because our erythrocytes, their main job is carrying gases around the body. They're doing this through diffusion. Okay, so we want a large surface area for them to absorb and release gases. We want those gases to come into contact with the cell and get into it quite easily. So their diameter, if you measure them across, is about 8 micrometers. Thickness is about 2 micrometers. They're pretty small. Okay. So that membrane on the outside of this biconcave disc is kind of flexible. So you can bend these cells around. That's also helpful for making sure that they can move to where they need to be. If we look inside these cells, part of why they're able to be so small is that they're missing a lot of stuff. So once we get to mature red blood cells, our red blood cells aren't going to like do mitosis and divide independently from mature red blood cells. We're actually going to need to go through kind of that whole flow chart to get a new red blood cell, that whole progression of different types of cells. Okay. This is because we don't have a nucleus, so we don't have that full DNA genetic code kind of lying in a nucleus. So we don't have that machinery to fully duplicate our red blood cells. And we don't even really have organelles in our erythrocytes. We're not bothering with mitochondria. We're not even doing glycolysis, right? So not doing our effective form of ATP production, but we're not even really doing much glycolysis. There's not much function going on here from an ATP perspective. Their major job is really just to move O2 and CO2 around, and we gave them all the components that they need to do that job earlier on, while they still had a nucleus, while they still had organelles in some of those earlier types of cells that you saw in that little flow. Okay. So one of the things that we made previously and put into our red blood cells is a super important molecule called hemoglobin. So hemoglobin's big job that we focus on, is to move around oxygen. Okay, so we'll have a little bit of oxygen just kind of dissolved in our plasma. We'll notice that when we talk specifically about how our gases are moving around the body. But having hemoglobin really, really makes it possible to move a lot of oxygen. So about 98.5% of the oxygen you're moving around your body is bound to a hemoglobin molecule. So that's what this HB is. That just stands for hemoglobin. And only about 1.5% is directly dissolved in plasma. Okay. So when we look at the structure of a hemoglobin molecule, it's got like 4 pieces. It has 4 subunits that we should say. So when we talked about levels of structure in protein, right, we were building amino acids into chains, polypeptides. Then we had layers of folding, right? So we would have had one polypeptide folding into each of 4 subunits. So we can see 1, 2, 3, 4. Okay. And in the future, when we talk about hemoglobin, we're going to really draw it as just 4 boxes. So a little square, little windows in it. When we think about how much oxygen we can put in a hemoglobin molecule, that's because within each of these subunits, we've got a globin part. So the globin part here are all those red lines. That's basically the protein part of the hemoglobin. And in the center, we have heme groups. So these heme groups, these little flower things here, those are what the oxygen is actually binding to. So you have one in the center of each of these subunits, and they, if we pull them out, have iron at the center of them, which we're pointing out in part because that iron is going to be something that we're going to need for adequate levels of hemoglobin, for adequate levels of gas transport as we start to think about that. Okay. So not all your hemoglobin has oxygen bound to it, and definitely not all your hemoglobin is totally full of oxygen. All right, and we'll talk about what I mean by full, but right, you can imagine I told you we're going to draw it as a square. Right, so later on, we're going to be talking about how many of these 4 spots are occupied at any given point in time. Okay. But if we're fully oxygenated, our hemoglobin is going to be bright red in color. If we're deoxygenated, we see a slight color change. Hemoglobin is going to be a darker red, which is why the veins, when you look at them in lab, look kind of dark partially because they got thin walls. They've got this deoxygenated dark red blood in them. Okay. So the main job of hemoglobin, our main focus of hemoglobin, is thinking about how oxygen binds to those heme groups and cycles around the body. It is the case that hemoglobin does also help us carry some other things, not at those heme sites, so kind of elsewhere on our molecules, but we're going to see that hemoglobin also binds a little bit to carbon dioxide, some hydrogen ions, and dangerously to carbon monoxide. That's not an error there, right? Carbon monoxide poisoning is real, and it's because of how carbon monoxide actually kind of competes with oxygen for some of these slots. Okay. But for now, hemoglobin's in red blood cells. We got 4 subunits. We got globin parts. We got 4 heme groups in total because they're inside. So another thing you find in red blood cells, an enzyme called carbonic anhydrase. So this is also important when we're thinking about transporting gases, because carbon dioxide kind of lives here. Okay. So when you have carbonic anhydrase present, we can speed up this reversible reaction where we take carbon dioxide and water, and ultimately we're going to see turn it into carbonic acid, but then into protons, so hydrogen ions, and bicarbonate ions. Okay. So when I said that chemoreceptors measure carbon dioxide levels and carbon dioxide can mess with pH, you don't need to know about that for right now, but this is part of why carbon dioxide messes with pH. The H in pH is kind of for these hydrogen ions. Okay, so we're going to see that this ability to convert carbon dioxide and water into these different types of ions and back is important for maintaining acid-base balance in the blood and for transporting carbon dioxide around. We have to think about hydrogen ions and bicarbonate when we think about carbon dioxide and what's going on with our blood as a whole big picture. Okay. So we've got this enzyme, carbonic anhydrase, in our red blood cells as well. So they don't have DNA, they actually also kind of don't have RNA, they don't have organelles. How do we get these erythrocytes, these red blood cells, right? They can't divide on their own because they just don't have the machinery to do it. They don't even have the genetic code to build a new red blood cell. And the answer is that we're kind of always filtering out our red blood cells, killing them off, recycling their parts, and then producing new ones from these cells that have the potential to divide into many different types of blood cells that we're going to store in our bone marrow. So the lifespan of any given erythrocyte, any given red blood cell, is about 120 days. So that's about how long they can function before they start to get too damaged, start to become less effective, and we filter them out. So every second, you're replacing about 2 to 3 million red blood cells for a total of about 200 billion every day. Okay. So we're filtering them out, mainly at the spleen, although we'll see the involvement of the liver as well as we step through the whole life cycle of a red blood cell. So that's where they're kind of dying. Where they're coming from, where they're being born, is in that red bone marrow through a process called erythropoiesis. So erythro, because these are erythrocytes, are red blood cells. Poiesis, the Greek root, I assume it must mean like creation or something, but I haven't looked that one specifically up. Okay. But we are going to notice those parts of the word because our hormone that is in charge of this has almost exactly the same name. So erythropoiesis happens through this molecule. Erythropoietin is going to be our signal for making more red blood cells. So erythropoietin is a type of hematopoietic growth factor. So like making blood, so heme from those heme groups for blood. Poietic must mean something about making stuff. Growth factor for growing those blood cells. Okay. So this molecule comes from the kidneys, and the kidneys know that you need more red blood cells when they start to see dropping levels of oxygen, maybe changes to blood cell through the kidneys. We're going to see that the renal system, the kidneys, is really involved in a lot of kind of management of what's in the blood. Okay. So our kidneys produce this chemical erythropoietin and tell us we need more red blood cells. And so this is a trigger for stem cells in the red bone marrow that lies inside your bones. So ever chop a bone in half and you can see it's kind of hollow in the middle, right? That's where the red bone marrow would go. So we've got these stem cells hanging out inside your bones when we have erythropoietin come through. So it's coming through in blood. There are actually blood vessels that go into your bones. We know that it's time to make new red blood cells from those stem cells. Okay. So we go through that flow of different types of cells, starting with our hematopoietic stem cells, and then producing hemoglobin, producing many kind of progenitor cells, so many of our earlier cell types that we have to go through, developing them. Once they have enough hemoglobin, they have their carbonic anhydrase, they have the stuff they need. They lose their nuclei, they lose their organelles, and then migrate out of the bone. So you shouldn't see red blood cells or any of those earlier cells in our kind of flow of differentiation. So differentiation is just kind of becoming different, so making different types of cells. Okay. So you shouldn't see any erythrocytes that have nuclei and organelles. The earlier step, right before our mature erythrocytes, is something called a reticulocyte. Sometimes in certain pathological conditions, that's something they'll test for in your blood. Like, did you have any of those cells kind of come out of the bone without losing the nuclei and organelles? And that can be kind of a problem. Okay. So in order to make a red blood cell, in order to make an erythrocyte, we're going to need some stuff. We need to make hemoglobin. We've got the globin part, that's the protein part, and we got the heme part in the center that is actively involved in carrying the oxygen. The heme part has an iron in the center, has some iron in the center. So we need iron in order to make red blood cells. There's not a lot of point of making red blood cells if you can't put hemoglobin in them. Normal content of hemoglobin in your blood, somewhere around, let's ballpark like 15-ish grams per deciliter. There's a typical range for men, for women. A little bit of variation across the board is kind of normal. Okay. But we've got a ballpark range we expect. So you're going to need to get iron from your diet, right? If you're like anemic, you may be told to eat more iron. This is part of why. You want enough heme groups, enough iron in your blood to carry around adequate oxygen. We also need folic acid and vitamin B12. These are necessary in general for making new cells. So when we say cell proliferation, right? In this case, we're trying to make new red blood cells. You also see these commonly in like pregnancy supplements because you're making lots of new cells if you're pregnant. So kind of important every time you're replicating DNA and making new cells. If you're making a new red blood cell, you definitely need this. New red blood cells are new cells. So you're going to need to eat those too. All right. We are going to talk about the steps of filtering out those erythrocytes and destroying them, partially because this is an A&P class and we need to know all the things your body does. Also because when we're thinking about that iron, we're going to see that some of the iron in the heme groups is getting recycled, and we want to understand how that's happening, where that's happening. So our end point of the life of a red blood cell that has been swimming around your body is going to be in the spleen. So it has a lot of blood flow to it, and it's filtering out old, breaking down erythrocytes. And then we're going to have the liver kind of help out here, because the liver is going to metabolize some byproducts as we break down those erythrocytes. So the spleen is going to grab them out of the blood, and the liver is going to help kind of pull them apart. All right. Part of why the spleen can do that is the spleen has macrophages in it. So that would be one of our type of leukocytes, one of our types of white blood cells. Some of them are clustered in the spleen. And what they're doing there is they're doing phagocytosis, so basically eating up other cells. Specifically, they're eating up old, fragile red blood cells that aren't functioning very well anymore. As this is happening, we're going to catabolize our hemoglobin, so break it down, right? Catabolism. So we're going to want to remove that iron because we're going to try to recycle that iron. And that's going to leave us with the rest of the heme group. The rest of the heme group is something called bilirubin. The bilirubin is going to go into your blood, and we're going to break that bilirubin down further. That part's happening in the liver. And the result is that we're going to have bile being made in the liver as it's breaking down these red blood cells, this bilirubin. That bile goes into your intestinal tract. It's bright green at first, but when it comes out, this is why feces are brown. And some of the rest of it is going to be released into our bloodstream and end up in the urine. Okay. So the iron is part of the really important bit here. The iron is what we kind of want to focus on when next time we talk through the life cycle of the red blood cell, because we need it for new hemoglobin. So we're recycling it. We're getting some of it through dietary sources, and we're going to need to be able to transport it between these various organs and the blood in order to make new red blood cells. So we'll stop here, and I'll see you all next time.